Hello, good afternoon, everyone. I'd like to welcome you to the 2017 Alan S. Michaels Lectureship. This is a very important lectureship for us because it honors one of the fathers of biotechnology in our department. And this lectureship, which was begun 22 years ago, has served as a forum for distinguished engineers and scientists in academia and in industry. It allows them to share their views on research and development in biomedical engineering and bioengineering. Now, before introducing today's speaker, I'd like to say a few words about Alan Michaels. Alan Michaels was a very important part of the department in MIT's life, starting in the 1940s when he got his bachelor's degree in 1944, his MSCEP in the Chemical Engineering Practice School in 1947, and his SCD in 1948, all of them in chemical engineering. Now, subsequently, he joined the MIT faculty for a couple of years and worked with Doc Lewis and E. Hauser in teaching colloid chemistry. He took a brief break, and then after a year in industry, he came back in 1950 to assume the position as the co-director of the Soil Stabilization Laboratory. Now, Alan Michaels' research interests at MIT were innovative and diverse, and they included everything from colloidal phenomena to soil mechanics and polymer permeability and its dependence upon polymer structure. What I think is one of the most fascinating areas that Alan Michaels examined were the interactions between polyelectrolytes and the generation of complex coacervates from charged polymer systems, which led to the birth of a range of new areas involving polyelectrolyte systems as material sets themselves. He had a particular interest in perm-selective polymeric membranes, which led to the current great interest in membrane separation processes in the chemical process industry. And in 1962, Alan founded Amicon Corporation, which is now a part of Merck Millipore, which produces filtration materials for separation processes, the biotech and healthcare industries. He left MIT back in 1966 to serve as Amicon's president. So here you have a slightly different view of Alan Michaels. You had the more sober, traditional photograph, and here he is enjoying himself a little bit later in life. Alan, in fact, received many honors and awards during his life, including election as a founding fellow of the American Institute of Medical and Biological Engineering, which AIMBE, which is one of the key honorary societies for chemical and biological engineers in this field. In addition, he's had several distinguished academic appointments as well as industrial scientific advisor positions for the food, pharmaceutical, and chemical industries. Alan Michaels was a pioneer in the application of chemical engineering principles to bioengineering. His leadership in forging cross-disciplinary collaboration between the medical and life sciences and chemical engineering has been recognized by our lectureship today, the Alan S. Michaels Distinguished Lectureship in Medical and Biological Engineering. In the spirit of the translational efforts in bioengineering that Alan represented, we're very honored to have Professor James Lau as our 2017 Alan S. Michaels Lecturer. Professor Lau received his bachelor's degree in 1980 from National Taiwan University and his PhD in 1987 from University of Wisconsin-Madison, a great chemical engineering department. After working as a research scientist at Eastman Kodak at Texas, he started his academic career at Texas A&M, another great chemical engineering department, and then moved on in 1997 to UCLA, and where he served as department head most recently up to 2016. Since June 2016, Jim has actually been on leave from UCLA to serve as the president of Academia Sinica in Taiwan, which is actually the National Academy of the Republic of China. It supports research activities in a wide variety of disciplines, ranging from the mathematical and physical sciences, to life sciences, to humanities and social sciences. As an educational institute, it provides PhD training and scholarship through its English language Taiwan International Graduate Program in Biology, Agriculture, Chemistry, Physics, Informatics, and Earth and Environmental Sciences. It's interesting to note that Academia Sinica reports directly to the president, and therefore enjoys autonomy in formulating its own research objectives. In addition to academic research on various subjects in the sciences and humanities, Academia Sinica's major tasks also include providing guidelines, channels of coordination, and incentives with a view to raising academic standards in the country. Professor Lau is an elected member of the U.S. National Academy of Sciences, the National Academy of Engineering, and academician of Academia Sinica in Taiwan. He also received numerous awards and recognitions, including the Presidential Green Chemistry Challenge Award in 2010, the White House Champion of Change for Innovations in Renewable Energy in 2012, and the Eni Renewable Energy Prize bestowed by the President of Italy in 2013, as well as the 2014 National Academy of Sciences Award for the Industrial Application of Science. Jim Lau's research has focused on metabolism, including its biochemistry, regulation, and redesign. He uses metabolic engineering, synthetic biology, and systems biology to construct microorganisms to produce next-generation biofuels and to study the obesity problem in humans, a very broad and diverse range of applications. Dr. Lau and his team also developed mathematical tools for investigating metabolism and guiding engineering design. Currently, their main projects include engineering proteins and biochemical pathways for CO2 fixation and production of fuels and chemicals. The ultimate goal is to use biochemical methods to replace petroleum processing and to treat metabolic disease. His talk is entitled Redesign of Metabolism for Carbon Management. Let's welcome Dr. Lau.
And thank you, Paula, for the wonderful introduction. And thank you for inviting me to be the 2017 Michaels Lecturer. As we know that Adam Michaels was a legendary person in chemical engineering, and many of us have benefited from his achievement, both in academia as well as in daily life. Many of you probably don't know that he was one of the pioneers in drug delivery. And today, drug delivery is such a common practice in the pharmaceutical companies. And back in 1960s, 1970s, that was a novelty item. So it was indeed a great honor for me to join the long list of very distinguished previous lecturers, Adam Michaels lectures. So thank you very much again. As Paula said, I took a leave of absence from UCLA about a year ago to head up Academia Sinica, which is the highest research institute in Taiwan. It has actually many components, total about 8,000 people with 1,000 PIs divided into 3 divisions. Humanities and social sciences, math and physical sciences, as well as life sciences. You can see this is the campus, and we are developing a biotechnology research park around this area, which is supposed to be finished by the end of this year. It will house about 60 startup biotech companies trying to mimic the Kendall Squared atmosphere to create an ecosystem for innovation in biotech areas. So here are some pictures of our campus. It actually, okay. On a good day, it looks pretty. I invite you to visit Academia Sinica if you come to Taiwan. So today I'm going to talk about carbon management. And of course, we know that the most important problem that we're facing in terms of carbon is carbon dioxide. A few years ago, I remember when I looked up this number, the carbon dioxide concentration in the atmosphere was about 300. And when I was in high school, that was 260 something, long time ago. Now it's exceeding 400 ppm. So why does that matter? If we have carbon dioxide concentration over 400, over 500, it is predicted that the average temperature on the surface will rise above 2 degrees Celsius, more than the pre-industrial level. Now, we are already 1.25 degrees Celsius. Okay, so we have very little margin to go. And this is a rising trend. We don't see any slowing down. People may argue the consequence of this, but I don't think few people can argue that the CO2 buildup is a fact. Okay. And regardless of the exact consequence of CO2 buildup, at least most people will agree that we cannot take the risk. Okay, we know also for a fact that CO2 is a greenhouse gas, and not just CO2, but also other kind of C1 carbon, for example, methane, which is also one of the compounds that I'll be talking about. So let's take a chemical engineering standpoint, do a balance around CO2 in the atmosphere. What's the input, what's the output? So the output of CO2 from atmosphere on land is the photosynthesis. It turns out that every year we took out 120 gigatons of carbon. This is only carbon from atmosphere. And half of it is immediately returned back to the atmosphere by respiration. The remaining parts will be deposited in soil and litter, slowly released back. A much more complex system happened in the ocean, and it's not very well established. But still have a lot of uncertainty. But regardless of the details of this balance, this system was destroyed after Industrial Revolution, when we start to dig out carbon from underground and burn it to CO2 released into the atmosphere. The fossil fuel we use put out about 8 gigatons a year, and it's rising. So this process cannot be absorbed by natural processes, and that caused a very rapid increase in CO2 concentration. So what are the solutions? Obviously, we dig out less. So instead of using fossil fuel, we can use solar or wind power as an energy source, but that would take some time to replace everything. Also, we can do biological ways. We can try to take CO2 from the atmosphere. First, fix it to something else, and then try to convert that to fuel. And of course, sooner or later, we'll burn back to CO2. And notice that biofuel will make easiest argument if we utilize CO2. If we utilize any of the existing fixed carbon, what we are doing is largely speed up the CO2 release. Okay? So this subtle point will make the biggest argument that we have to focus on CO2 fixation, no matter what you do. Okay? We have to take CO2 from the atmosphere, no matter what you want to use it. You want to use that to replace the fossil fuel or use that as a material or do anything. So CO2 fixation is important. Another argument is that even if we forget about the fuel, we can replace all the fuel someday. I say someday, and it may not be right away. If we can replace all this fuel with electricity, with the solar electricity and so on and so forth, we are still exceeding the limit that I showed up in the past slide. Okay, we will still need to know how to fix carbon dioxide to retain the previous balance. Another problem we're seeing is a lot of landfill gas, methane, in addition to natural gas that we found that leak out from the underground. That is at least 25 times worse than CO2 as a greenhouse gas. Okay? So we'll need to learn how to manage methane. Okay, of course, we can use methane to do some biotransformation, but that's even more difficult. So these are the problems, the long-term problems that we're dealing with. And we try to solve this problem using biological ways. So let's look at what biology offers. So this is a composite slide of most of the biological transformations that has been evolved. First, we have the photosystem that harvests sunlight, converts light energy to biologically available energy in terms of ATP or electron carriers that can drive CO2 fixation, and largely through the Calvin cycle or the CPP pathway. This Calvin cycle reduces CO2 to glucose or intermediate three carbon compounds stored in cellulose or other biomass. When we want to utilize this biomass, we then reverse this process to make various kinds of compounds. And notice that regardless of what you want to make, there are two intermediates that are very important. Almost whatever you do, you have to go through these two intermediates. One is called the pyruvate, which has 3 carbons. The other one is acetyl-CoA, which has 2 usable carbons. And so in between converting 3 carbon to 2 carbon, you lose 1 carbon. And that limits dramatically the yield, the carbon yield of any biological processes to 66.7%. So we'll deal with that later. First, let's find out what kind of compounds that we can produce using biological ways. Of course, the easiest way is to use these compounds, this sugar, to make ethanol, okay, which human beings have been enjoying for thousands of years. In addition to that, we can also think of more advanced fuels. A few years ago, we put together a series of pathways that allow us to extend the biochemical systems to make longer-chain alcohols, for example, isobutanol, okay? Or 2-methyl-1-butanol, 1-butanol, and so on and so forth, and you can keep extending this chain length. And this isobutanol probably is the best studied compound and has been commercialized today. And since then, our lab, and together with many other labs, and at least 2 in MIT, Greg's and Chris' lab, have shown that we can practically make just about every compound that we can put together on the drawing board. The challenge is, can we make it efficiently? So the chemical synthesis, replacing chemical synthesis using biological ways is not a dream anymore. It is in practice that it is possible. Now, can we think about more fundamental changes that allow us to utilize CO2 directly? An easy solution is, why don't we utilize an organism, for example, cyanobacteria, which can fix CO2 with the help of sunlight. And if we can steal its intermediates using metabolic engineering, then we can directly convert CO2 to any of these compounds that we are interested in. And we did exactly that and showed that we can produce quite a bit of different kinds of compounds with the help of sunlight directly from CO2. And this is an example of making isobutanol. Of course, the titer is still too low. And one of the difficulties in using this type of approaches is that it requires sunlight. It requires a lot of surface area. And although there are many other difficulties involved. And this problem probably is one of the bottlenecks that prevented this commercialization. So let's look at the whole system. We try to utilize sunlight and first separate the charge, then use the charge to fix carbon dioxide to produce biomass, and then use biochemical ways to convert biofuel, either in one cell or in many different steps, in plants, in bacteria, and so on and so forth. But let's look at the efficiency. The best efficiency from sunlight to biomass have been calculated to be no more than 4.6% or 6%. This is theoretical, okay? From C3 plants or C4 plants, the 2 types of plants, C4 is more efficient, C3 is less efficient. But in practice, people never seen anything greater than 1% in terms of efficiency from sunlight to biomass. However, if we look at photovoltaic cells, which can separate charge from sunlight to electricity, the efficiency is on average 15%. No matter what you buy from the street, it's probably 15%. If you are willing to spend a little bit more money, you can exceed 20% or even 30%, or you can build one exceeding the record of 43% or something. It's very dramatic. The problem is that this charge cannot be used to fix CO2. It cannot do any chemical reaction at this complexity. So if we look at the biological processes, if you remember your high school biology, there are two parts. One is called light reactions. The other one is dark reactions. What that means is that the light reactions depend on light, this converting solar energy to ATP, the energy currency that biology can use, or NADPH. This is the electron carrier. And the dark reactions are the parts that actually fix CO2. And these two systems don't need to be together. If we can separate these two systems and replace the light reactions with the man-made system using photovoltaics, get to electricity. Unfortunately, electricity cannot be directly utilized by any living organisms, at least most of the living organisms. So we have to convert electricity to something that living organisms can use. So the easiest way is to use hydrogen, that we can use electrolysis to split water to make hydrogen. Okay. Of course, hydrogen comes with its own issues. So our alternative is to use formic acid, which can also be made by electrolysis in the presence of CO2. Of course, you need to select the appropriate catalyst on the electrode surface. You can make formic acid. And then now formic acid has 100% solubility in water. You don't have the mass transfer problem that you deal with in hydrogen, and much less of a hazard issue with hydrogen. So if we can do that, then we can use formic acid as an electron carrier to drive Calvin cycle to reduce CO2 to something. All we have to do is find the organisms that can utilize formic acid and engineer its pathway to produce the kind of fuel that we are interested in. If we do so, we can increase the theoretical yield dramatically, although in practice we're still way below. So we pick an organism called Rostonia eutropha. We engineer its metabolism, trying to steer its intermediates to make the chemical that we are interested in, which is isobutanol. And we first show that this organism now can convert hydrogen and CO2, or formic acid and CO2, to the kind of compound we're interested in, isobutanol and 3-methylbutanol and so on and so forth. The next, we then have to put this together with the electrolysis system so we can directly utilize sunlight to drive this CO2 fixation. And indeed, we accomplished that. So we have a system where it has two electrodes that upon the electricity introduction, we can fix CO2 to formic acid and then use the microbial system to convert formic acid to isobutanol. So this is the first demonstration that we can fix CO2 with the help of electricity. So this electricity either comes from solar panel or from wind power or any city grid. So this type of approach is not so much for the purpose of making biofuel per se, but it's a very good approach to store electrical energy. As we know, there's very little alternatives to store electricity, okay, other than batteries. But battery has one major problem, that's the energy density is too low. Okay? And of course, although it's improving dramatically, but if we can store the electrical energy into carbon bound and not just limit to hydrogen. The current idea is that let's store the electrical energy to hydrogen, which is a gas. Okay? So you turn a battery into a gas battery, a hydrogen. If we can at least make that into a liquid, the density is higher. So at least this type of approach deserves a lot more attention than it has received so far. So that is our effort of trying to utilize this type of scheme, try to maximize what we can do using available infrastructure provided by biochemistry. But as I alluded to earlier, there's a major problem of this type of approach. That is, when we utilize this carbon fixed by Kelvin cycle, we will have to first convert to pyruvate and then to acetyl-CoA. In the process, we lose 1/3 of carbon. And this is the major limitation of all biological processes, which I call a glass ceiling. So if you look at the mass yield, if we try to make ethanol from glucose, the maximum theoretical yield you can achieve is about 50%. And this yield goes down if you increase the chain length, because you have to do this cycle more and more times. Every cycle you go through this pyruvate, you lose 1/3. Every time you lose 1/3. So the mass yield goes down. Although the energy content is about the same, unfortunately, we sell this biofuel based on its weight rather than based on its energy content. So if you then put together the big picture, ethanol, I don't know the current price, but at the time I made this slide, it's about 80 cents per kilo. Sugar is about 40 cents per kilo, but you need 2 kilos of sugar to make 1 kilo of ethanol. That's why it caused such a big problem, such a big difficulty in making biofuel economical. So let's look at why we have to lose CO2. It all happens in this process, pyruvate to acetyl-CoA. 3 carbon to 2 carbon, it loses CO2. You then ask, why does a cell want to lose CO2? Because it wants to derive electrons from that. Okay? Because the cell needs to grow, so it rather burns part of the molecule to CO2, derive the energy in order to grow. Okay, that's for the purpose of growth. But if we want to conserve CO2, can we do something? Can we try to conserve CO2 without deriving all the electrons? Chemically, it's possible. You can write down the chemical balance. You can get 3 moles of C2 from 1 mole of C6. It's completely chemically balanced. Then how do we do that? A few years ago, we designed a process that depends on an enzyme called phosphokedolase. This enzyme was discovered about 50 years ago. It can do a simple reaction, split 6 carbon to 2 and 4, or 5 carbon to 2 and 3. You don't need to worry about the detailed chemistry. It's 6 split to 2 and 4, or 5 to 2 and 3. So you got 2. The problem is, how do you deal with the residual 3 or 4? So here, being a chemical engineering group, we had the advantage of learning the traditional chemical engineering principle, which is recycling. Okay? Chemical engineers know how to recycle. So we can recycle this E4P, trying to recycle that back to 6 carbon. If we can do that, then we can complete the balance. So how do we do this recycling? It involves a series of chemical rearrangement, which we call the carbon rearrangement. And if we can do in a fashion that does not require any redox or any ATP, then we can recycle this for free. And if you look at the chemical structure, indeed, we can do that. Okay, the chemical the redox state of E4P and F6P are identical. So you don't need any electrons in there. So number 1 is satisfied. The energy is about the same as well. So theoretically, you can do that. And in practice, how do you do that? So we designed a complex scheme. Again, a lot of loops. 3 molecules of E4P comes into the system, which we vaguely call the system, involve many different enzymes. We can rearrange this carbon. The output is 2 moles of F6P. The enzymes involved are all well characterized in pentose pathway, in glycolysis, in gluconeogenesis. These are the enzymes that you learn in Biochemistry 101. Okay, and you forget immediately after that, after the final exam. So I forced the student to look at the carbon migration, and you can even follow the carbon migration exactly where it goes. Okay? And you can prove to yourself that this works. And if we put in another way, you can then convert a 6 carbon to 3 molecules of 2 carbon, or 6 carbon to 3 molecules of 2 carbon using many different ways. Okay? This scheme utilized a split between 5 to 3 and 2. This scheme utilized 6 to 2 and 4. And these two modes can recombine and get infinite variations. So these are the possibilities. So you can draw that easily on the board. Can we actually do that? Well, the first part, the chemistry looks fine. How about thermodynamics? Okay, we did a thermodynamic calculation. The traditional pathway is EMP, Embden-Meyerhof pathway. It looks like this, one way down to lactic acid. Now, if we use what we call the non-oxidative glycolysis, it doesn't involve any oxidation because we didn't derive any electrons. It's also one way down and one big driving force in between. So theoretically, it is possible as well. So how do we demonstrate it? We first put together this system in vitro, probably 10, 15 enzymes, put in a test tube, optimize it, and demonstrate that we can work. And then we cloned all these pathways, all these enzymes into E. coli, knock out all the pathways that are competing with it, and we're able to demonstrate that, indeed, we can convert a 5-carbon sugar, this is xylose, to 2.5 moles of C2 compound. So almost theoretical yield. If you leave out any of the enzymes, the yield dramatically dropped. So all these lower bars, those are controls. And we can do a detailed kinetics and show that, yes, we get the ACP, the acidyl phosphate, with the theoretical yield. So we first demonstrated that this type of work, this type of design, although artificial, can actually work in E. coli. But it has a problem. What is the problem? Because I mentioned earlier, the reason why the cell wanted to do this partial oxidation is to derive energy, derive electrons out of this. Now we take that away. We conserve carbon, that means we cannot derive any electron out of that. How does the cell grow? Indeed, the cell cannot grow. So we have to solve this problem. To design completely the whole system so that the cell can grow with this NOG. Okay. How do we deal with that? We first convert glucose to acetyl-CoA. Okay. And we can do that in a fashion that's completely carbon balanced. Okay. Get the 3-acetyl-CoA. Then we will have to convert acetyl-CoA to C3 again. Okay. This requires another engineering of the lower part of the central metabolism. It requires a TCA cycle rearrangement. So normally, what we do in the cell, in almost every organism, we first make C3, from C6 make to C3, then C2. Okay? From C6 to C3 is very efficient, but it gets to C2, then it loses CO2. So our new design, we are very efficient in making C3, but we are not very efficient, very efficient making C2, but not efficient making C3. There's a trade-off. This is a native way, is supposed to grow well. In our way, it doesn't grow as well, but at least it can grow. However, if you want to derive intermediates, acetyl-CoA for production, this may be a better way. So we designed this pathway. Of course, the first 100 times we tried failed. So it took us a long time. Now we're able to show that we have a novel strain of E. coli that does not require TCA, does not require glycolysis. So this strain, what I did not mention, we had to knock out completely the lower part of glycolysis. So it cannot do the typical glycolysis. And we show that there's no other way that it can grow based on making C3 pyruvate than C2. Then only then and we show that we have a novel pathway that goes to C2, then C3. And this strand, as predicted, it grows slightly slower than the wild type. Okay? If we leave out the key enzyme, then it doesn't grow. Also, as predicted, this strand does not grow in the absence of oxygen because it cannot do fermentation. Without oxygen, it's completely dead, and this is exactly what we predicted. How does this guy behave? Okay. Normal cells produce mainly lactate as a byproduct, and this is our control using the plasmid that we have, but we stuck into the wild type cell. It doesn't work. And our new cells, after several generations of evolution, now we show that the main product is acetate, okay? And we get to the ratio of 2.4 per mole of glucose, okay? We still have byproducts in succinate, but that's easy to deal with, or if we can do some further engineering. So basically, now we have a novel organism that doesn't depend on glycolysis. In fact, we are currently proved that it probably doesn't depend on the traditional TCA cycle as well. So this is a completely, what we call a different way of life, okay? As opposed to what other people are trying to design a synthetic cell, which using synthetic way to deliver DNA, but play exactly the same music. The music here is different, okay? So moving on, if we have this Calvin cycle, okay, in any plant or cyanobacteria, we fix 3 carbon, only to lose 1 more, okay, to make acetyl-CoA. That is the problem that we set out to fix. Now, if we combine this with NOG, non-oxidative glycolysis, we can make 1.5 moles of acetyl-CoA from 3 moles of CO2. Isn't that great? We know that fixing carbon is a very difficult thing. Plant or cyanobacteria utilize an enzyme called Rubisco, which is notoriously inefficient. So if we can increase the efficiency per turnaround by 50%, that's great. So we look into this possibility, trying to see, can we do this in cyanobacteria? And before we do that, my students already told me that cyanobacteria has all the enzymes that we need for NOG. So the first hypothesis that we have is that maybe the cell already doing this, it's just that we did not know. And we looked through all the data bank. Indeed, every single cyanobacteria has every enzyme we need for this. So we are very excited. Let's try to artificially demonstrate that. So we put that in cyanobacteria. Unfortunately, the cyanobacteria die right away. This is normal cyanobacteria grow green, but we put in this NOG, it dies immediately. And that's not because of the toxicity that we introduced. We had control, we put in some sugar. We engineered the cell to allow the cell to utilize sugar, it can rescue at least partially. So it is not the toxicity of the compound. So we did further research. We look at this enzyme, phosphokedolase. It turns out this phosphokedolase is inhibited by ATP. So all these activity, the higher the ATP, the lower the activity. So this was interesting. Okay? Why would this guy want it to be inhibited by ATP? So again, we did a lot of survey. Okay? Pick many different enzymes from different cyanobacteria. These are all cyanobacteria. Every one of them is inhibited by ATP. But the same enzyme in other bacteria, these are in non-cyanobacteria. Many other bacteria have this phosphokedolase. These non-cyanobacterial enzymes are not inhibited by ATP except this one. Okay. So again, we see some interesting behavior. In addition, we see that it's activated by ADP, but not everybody is activated by ADP. Okay. Only the ones that fix nitrogen. So we begin to see some pattern. This guy is inhibited by ATP, activated by ADP, and they all appear in cyanobacteria fixing nitrogen. So why is that? And then we further characterize this. This guy is actually produced in a light condition. And yet, once we activate that, it stops. Okay? So our current hypothesis is that this is not the theory that we're trying to prove. This is not the cell trying to conserve carbon. Instead, this is a break. This is a break to CO2 fixation. Once the cell senses that you have not enough ATP, you take away the intermediates of CPV cycle to shunt that to acetate to make ATP instead of fixing carbon dioxide. Okay. So this is a surprise sidetrack that we discovered that even though nature has this, but it never get it to work for the good reason. It utilized for other things. So now it brings back to our original question. We wanted to keep this carbon fixed from CO2, okay? How can we keep this to C2? We cannot use this NOG because it becomes a break from native organism. So we have to design something else. So we design another one. This cycle, which we call reverse dioxylate shunt glycerate, is quite long. I'll tell you, you probably don't worry about the history of why we call this. In short, it involves a reverse dioxylate shunt. A reverse dioxylate shunt means typically a dioxylate shunt goes from dioxylate to, this is C2 to C4. Now we reverse this. Why is that needed? Because this is the only way that we can utilize another CO2 fixing enzyme, that's the PPC. Again, most of you probably are not familiar with these names, but this cycle allows us to take C3 into C2 in a stoichiometric fashion. So if you have everything balanced, you can have a C3 plus C1 to become 2 moles of C2. Okay? Or if you have glucose, you can split into even 4 C2 by fixing 1 CO2. So this cycle, the advantage of this cycle is that it is completely compatible with Calvin cycle. And also it allows us to fix 1 more carbon. In addition, there's something called photorespiration in plants, meaning that it produces a C2 compound called glycolate, which cannot be used. Okay? Many of the plants suffer from this problem, so the efficiency is low. And we designed this pathway, can convert this, use this glycolate into acetyl-CoA to be used, completely balanced. And previous ways to deal with this still lose CO2. So this C2, you had 2 moles of C2 to become a useful C2 compound. It loses 2 C2. So it burns the glycolate away. And this method allows us to recycle glycolate completely into acetyl-CoA. And so we test this idea in E. coli before going to plants. And we can demonstrate that we have this system, okay? Again, don't worry about all these different data. These are different controls. The only one number that I want you to focus on is this final number. Is what, 2.9, okay? Is the ratio between sugar, C6 and C2. So anything above 2, we're happy, okay? So this shows that we have achieved this, this carbon conservation, and followed by radioactive, not radioactive, the C13 label. Again, we demonstrate that we can achieve 2.7. This is directly from completely labeled the glucose and completely and bicarbonate. We can achieve greater than 2 compounds. Now we're ready to put it back into cyanobacteria to see how well it goes. So we put in cyanobacteria. This is CO2 fixation. We get a weird peak, and this peak turns out to be keto isocaprate. Okay, this is compound, the compound coming from the isobutyl pathway. the amino acid biosynthesis. We could not reason out easily why this is so until we look at the chemistry. This compound is produced from acetyl-CoA. So we have a system that we put in this acetyl-CoA synthesis. It probably, we have not completely proved that, it probably increased the acetyl-CoA supply, and therefore it comes out this byproduct. And if you look at the growth, it indeed grows better than its control. Okay? But notice that it only lasts for 4 days. After that, the toxicity drives in. So it's not completely performing what we wanted to do yet. But it shows that it is possible now to completely change the carbon management cycle, the infrastructure in the cell, regardless it's E. coli or cyanobacteria. Okay, so designing all these pathways, why is it useful? Okay, even if it's not useful right away, but where are we heading? We know that traditionally, if you want to make ethanol, if you want to make 3 moles of ethanol, you need 1.5 moles of glucose. Now, if you want to make 3 moles of ethanol, you only need 1 mole of glucose. You save 50% of sugar right there. However, it's not completely free. You need reducing power. Where do you get reducing power? Hydrogen. So if your hydrogen price, after some calculation, if your hydrogen price is lower than about $3 per kilo, then it probably makes sense. Okay? But of course, this is a function of sugar price and how you design the process and so on and so forth. So we can think of this comparing the NOG way versus the traditional way. What we have accomplished is a hydrogen-assisted CO2 fixation. We line up a CO2 fixation with hydrogen with a traditional fermentation that becomes our NOG. We use hydrogen to fix the CO2 that would otherwise be released. Again, where do you get the hydrogen? The best way to get hydrogen today is using steam reforming for methane. That can give us, if you get 1.5 moles of methane, it can give us the 6 moles of hydrogen that you would need. So together, you will produce 1.5 moles of CO2 if you want to produce 3 moles of ethanol. As compared to the traditional way, you will generate 3 moles of CO2. So you still save some CO2. Okay? And this methane steam reforming is a pretty well established process. So another way to look at this is that if you compare this is NOG plus steam reforming, this is what we accomplish. This is traditional fermentation. The difference is what we call the methane upgrading. Instead of using methane, you add half of CO2 to it. You get 1.5 methane, you add half of a CO2 to make it ethanol. So this is intriguing. This process has never been seen before. And in fact, it's not even thermodynamically feasible. If you look at the thermodynamics, delta G is greater than, much higher than 0. So how can we accomplish that? At least theoretically. Because we are utilizing the delta G generated from fermentation, which is wasted. We couple this big delta G, negative delta G, with this positive delta G. That's what we are doing. Okay? So we utilize this principle of one big downstream reaction to drive a halfway upstream reaction. Okay? So there's a lot of games you can play by doing this rearrangement. Speaking of methane, we all know that methane utilization has been a problem. Okay, the typical way is first make that into syngas, and then using syngas to chemically make that into methanol. However, methanol is equally difficult to utilize, although you can make that into gasoline using MTG process. It's a liquid, it's a little bit better. But ideally, we wanted to increase the chain length of this carbon. We wanted to do a simple reaction. Okay? Any high school student can write down this equation. 2 methanol to 1 ethanol, get rid of 1 water. This is a classical condensation reaction. Okay? Delta G is negative, or you can make 4 methanol to 1 butanol, get 3 water out. Again, delta G is negative. However, this cannot be done. Not chemically, not biologically. No chemist can do this. Okay, the closest reaction is this Gerber reaction from C3 coupled with, from methanol, C1 coupled with C3. This is the closest reaction. The commercial reaction is methanol to gasoline is coupling of methanol to longer chain non-specifically. Okay? That's happening under high temperature and high pressure. So if you want to do this specific reaction, then you are stuck. Okay? Chemistry cannot perform that. Biology, if you want to do this, there are a lot of organisms that can consume methanol. However, in the process, you lose CO2. Okay, as I said, all the problems again. So we have another challenge. We have to redesign this, to redesign the pathway so that we accomplish this condensation as well as conserve the energy. So we utilize the same principle of redesigning this pathway. We call this methanol condensation cycle. It's combining an NOG with a natural RUMP pathway. We call it methanol condensation cycle. Again, there are 2-3 modes of doing that. You can make the methanol, here we use formaldehyde as an example, to butanol, to acetyl-CoA, then to butanol, or to ethanol. Either way, it works. So we first demonstrate that in purify the enzyme. The same old trick, we put together all the enzymes, see actually it works the way we want it to work. And we label the methanol. In this case, we use formaldehyde. Okay? We can demonstrate that we can produce double-labeled ethanol. Okay? And if we leave out any one of the enzyme in the return cycle, we only get 1 label. So this proves that we have a catalytic cycle. Okay? Why this is important? Because people have demonstrated this type of work using, not exactly the same, but this line of work using the heterogeneous catalysis, only to find out later that it's not a catalytic cycle. So it's very important for us to demonstrate that we do have a catalytic cycle. And in vitro, we can actually demonstrate conversion of methanol to ethanol with a carbon yield of 80%. Not 100%, but not too bad. To butanol, it's about 50%. It's not too bad. So this system, like the NOG system, also has the same problem that NOG system we demonstrated. That is, the cell cannot grow because we conserve all the carbon. As soon as you conserve all the carbon, the cell cannot grow. So once the cell cannot grow, it's hard to deal with the cell. You cannot do the evolution. You cannot do many things. So we had to redesign this again. We had to redesign this cycle so that the cell can grow on methanol. So we did that. Okay? And I'm not going to tell you the details and not the time limitation. I still have one short story to tell. So we designed a similar system. So now we can allow the cell to grow on methanol and xylose together. Without methanol, it cannot grow. Okay, xylose only, it cannot grow. So we had to redesign the system, knock out a few genes. So normally E. coli can grow on xylose happily. But now, this cell has to have methanol together with xylose in order to grow. So this system allows us to do a lot of evolution that biology can take advantage of. We are further characterizing this system so that we can perform something what we call xylose upgrading. Okay. Xylose is also a very abundant sugar. Everybody knows that. And xylose utilization has been a problem as well. A few proposals come out there, but xylose is difficult to use. If we can utilize xylose and methanol together, C1 and C5 together, let's make C6 out of it, and then utilize the C6. Okay. So this is our idea of methanol, of xylose upgrading. Okay, so hopefully we solve two problems at the same time. My last problem, a short story I wanted to talk to you about is the obesity. So far we've been dealing with a carbon problem. We want to conserve carbon. That's for bacteria. We create a problem in the atmosphere. We want to conserve the carbon to utilize it. For human beings, we have the opposite problem. We have carbon accumulation problem, which we want to burn away. So the two problems seem to be opposite, but in fact, they share a lot of common features. And I'm sure everybody knows the problem of obesity. I'm not going to go through that. So the carbon in our bodies is mainly burned through fatty acid in the TCA cycle. First go to fatty acid and then beta oxidation to acetyl-CoA to TCA cycle. So what we wanted to do is create another channel for the cell to burn fatty acid, burn acetyl-CoA. Typically, acetyl-CoA is burned through this TCA cycle at the end. We cut through a TCA cycle. This is the dioxide shunt I've been talking about. And in our work before, we are reversing this cycle. Now we want to push it the normal way. And not only that, we push it even up here and go down. So we burn that into 2CO2. So if we can accomplish this, we create another channel for the cell to burn fatty acid. So this idea, we come up with this idea many years ago, but was not confident enough to push it through until I persuaded a very courageous graduate student to do so. However, we are still not very comfortable that this could work because the most likely result is that there's no change. Whatever you do, the cell just ignore it, or the organism. Or it could actually decrease the fatty acid because fatty acid oxidation, because it could come halfway and come out as the citrate, and then malonyl-CoA inhibit the fatty acid consumption. Or it can, another 2/3 way, it can go to glycolysis, go to gluconeogenesis and make more sugar out of your fatty acid, which is not what we wanted to see. Only a small chance that it can increase the fatty acid degradation. So after many, many trials and tried many things, and finally we were able to demonstrate that we can indeed decrease the weight gain if we feed the mouse in high-fat chow, high-fat diet. Okay. And so you know what to eat or what not to eat. And the controls mice gained the weight dramatically. The mice with our engineered pathway with additional fatty acid degradation, it gains much less weight. Okay? And of course, we did a lot of different controls to show that it is indeed burning the fatty acid the way we want, and we open up the stomach, and indeed that it produces much less fat in the body. So here are the different aspects of this carbon management. Okay? So you know that it is important for ranging from environmental problems to medical issues. It's all about carbon management. And we all relying on what nature gives us. And it's probably time that we think about how to redesign this infrastructure. We know, just like our nation, that the infrastructure is getting old. So it's time to redesign the new infrastructure. And this is just a few examples of demonstrating how we could redesign this carbon management infrastructure. So here are the people actually contribute to this work. There are so many people contributed to this line of work, and many of them have graduated. This is a composite picture of my group over many generations. Thank you very much for your attention, and I appreciate any questions.
Our speaker and present him with the presentation of this award for the lecture for L&S Michaels.
Thank you.
And thank you very much, Jim. Thank you for your beautiful lecture and for your presence here.
Thank you.
Open things up for questions. We have a roaming microphone, so please pause a minute while we get the microphone to you.
So in your LOD system, what happens to the Thank you for the great lecture. In your NOG system, what happens to the accumulation of acetyl phosphate?
Oh, acetyl phosphate goes to acetyl-CoA, okay, or acetate. Either way, it's fine, or you want to channel that to wherever you like. Okay. Typically, you first go to acetyl-CoA, then the acetyl-CoA, you can take care of it.
And, can you just add the enzyme that breaks down F6P to ACP? Can you just add it to normal E. coli? And can the normal E. coli accept that and makes more ACP?
Yeah, you can do a little bit of that, but it's not very effective. That's why we'll do a lot of major reconstruction, a lot of evolution to get it to work. The cell doesn't like ACP. ACP itself is toxic. Okay.
Thank you.
Thanks for the lecture. I just had a question about the reverse glyoxylate shunt glycerate system. Did you get higher product formation when you used cyanobacteria or E. coli?
Of course, E. coli is still performing better because we have much better control. Cyanobacteria is difficult to deal with, and we're just beginning to show some effect. But the potential is there.
Thanks for the talk. So E. coli, unless engineered, obviously can't make 3-acetate per glucose, but there are native organisms, acetogens, that can do that. They use the Wood-Lindahl pathway. So can you comment then on what the benefits of the engineered E. coli are versus one of those native organisms that already does that?
It's, of course, one would say, if you have one highway, why do you want to build another one, right? You're never satisfied with the only one you have. Okay. That's a general philosophical question. But specifically, the acetogens utilize a very complicated pathway. I'm sure you're aware of that, and it's just difficult to deal with. And so far, it's not well characterized. Maybe someday it can be characterized better. Maybe someday we learn how to deal with that. It's a different way to deal with it. And acetogens is fixing the carbon that it loses. Okay, instead of we try to conserve carbon, so a different approach. It may have different applications in different scenarios, but for now, one is easier to us, the other one is more difficult.
Jim, thanks a lot for your lecture. I learned a lot today. I'd like for a minute to ask you a question that goes to the methodological philosophy of the approach you described. I would call it an evolutionary, in the sense that you are making changes in E. coli and you are assessing the results. And then you make more changes to solve problems that they have showed up, like the interaction with the energy pathways you referred to. I am not in biotechnology in that spirit, but I'm a systems person. Back in 1985, we started work to automate through computers the synthesis of metabolic pathways. And that has been picked up in the recent years and has been growing significantly. So, in principle, one who could start from given substrates, go to desired products, and create through the computers an alternative of pathways, not only for the production. They could do that for the energy pathways. You can even go to the reproductive, the fatty acids and the vesicle formation, and even the amino acids and the nucleic acids for the whole cell. But let's stay with the production and the energy. So in principle, you can study the synthesis of many alternative combinations on the computer and assess them in terms of balance, carbon production, and in terms of thermodynamics. Then zoom the experimental work through the genetic modifications. And the technology is with us today. So my question is, why didn't you do it that way? Is it being done elsewhere extensively that way?
Indeed, that's exactly what most people are doing. And to answer your question, why it hasn't been more efficient, I'm sure many of people here in the lab, working in a lab, will know that it's much easier to draw on the board. It's another completely different story if you implement in vivo. Completely different story, because we just don't know the details. The kind of thing that we can draw, we can design are not detailed enough. Okay? Even though I show you the reactions, but they are by far a lot more. Then you ask, maybe we can do genome-wide, but those are the reactions that we know. Okay? Many of the reactions happen spontaneously that we don't know, or we know, but it's too difficult to control. Okay? Those are the difficult parts. Okay? In fact, every design that we put in, we have to test it in vivo. And most of the times, that if it works right away, we will celebrate. Okay? And most of the time, it doesn't work as designed, so we have to tinker through the many things and eventually get it to work.
And I appreciate that a lot. And I agree with you completely. Because what has been missing from those efforts is the element of regulation, of control, of sensing and controlling. And without it, the cell will not grow, will die. Therefore, you need to do what I described, okay, to include also the control mechanisms. And many of them are known, many of them are not known. But that is exactly the frontier, isn't it? Where one should be focusing attention.
Yes and no. Control is important, but since you bring up this topic, I would tell you a little bit of the way our philosophy and the way we're dealing with it. Control is one area, but I think robustness is even more important. Okay? The kind of thing that we design, we don't want to design something that requires perfect control. It only works in this condition, or the certain parameter, the enzyme has to be this high, or the Km value has to be exactly this. What if the cell mutate a little bit, then you die? So we try to design something that is so robust that it doesn't matter how much enzyme you put in, it works. Okay? And we are working on a few approaches to evaluate robustness using computational tools. And indeed, we see those issues. There are some designs that are more robust than others. For example, many of the designs that we put out did not work. Initially, we didn't know why. Because it's so easy to fail in the lab, so easy to fail. But if we go through our computation, we found that some designs are more robust than others. So what do we mean by robustness in this case? That is, it doesn't matter what parameter you have, you just put in the enzyme, it just worked. What do we mean by non-robust? You have to be specifically designed. This enzyme has to be 3 units. That enzyme has to be 5 units. And yet the Km need to be less than 4. Something like that, non-robust. Okay.
But for us in control, robustness comes always with the term control.
Okay, yeah.
So we cannot envision control systems without the term robustness.
Probably there's a valid point there, but we are only dealing with the pathway robustness. So here, and this is again philosophical. Okay? So here comes from a philosophy that I believe that before the quote-unquote control system was evolved in early days of life, there's no control. For example, this is philosophical, of course. You and I cannot prove that. Okay? In those days, how do cells grow? The cell has to grow in a very robust way. Okay? It, after millions of years, evolved the enzyme, boom, it's happy. Then another enzyme, happier, and then become a pathway, begin to grow. It has to be very happy, okay? Without any control, it has to grow, okay? Those are the type of system we want, okay? You first evolve the enzyme, okay? Just perform the chemical reaction, link them together, and then all of a sudden, it shows a benefit. They live together, and that's the beginning of evolution. And then after that, then you evolve a control system to regulate, perform better and better and better. So we're looking at the pre-control era, if you will. After what we accomplish here, I agree, we'll have to engineer the control, design the control, and all that kind of thing. But this is another philosophical point. Using our methodology, we examine all the native pathways. They're all very robust. That is, regardless of control or not, it just works. It may not work as well, but it works. Okay? And I think many of the systems you find out in bacteria, in E. coli, for example, the regulators you knock out, the cell doesn't matter. It grows. May not grow as well, but it grows. But the enzymes, you knock out the pathway, it dies. It's like in a highway, if you knock out a bridge, everything stops. If a traffic light breaks, doesn't matter. So it may not work very well, but people drives. Okay? So the kind of thing that we're trying to design is still very early on. Okay? We have to admit that we are not that sophisticated, okay? Because there's just so much we don't know about putting together hundreds of chemicals together, what will happen? And many of them involve reactive species. After all, we are doing chemical transformation at the same time. So what happened to the intermediates? Okay, they will react with each other. They'll react with enzymes. So we are working with a very reactive soup in here. Okay, it's not A to B. It's A star. That means very reactive. And so there are many, many reactive species in there that we don't know about. We know, but we didn't take care of it.
Are there any additional questions from the audience? Any questions? All right. My question is whether or not that traffic light analogy referred more to Boston or to Taiwan.
I think it's everywhere. It's everywhere. Yeah.
All right. Well, with that, let's thank our speaker one last time.
Thank you.